Hello, everyone. Welcome to today's webinar panel discussion on the latest annual Emerging Trends to Watch publication from CAS. I am Janet Sasso, an information scientist here at CAS, and I will be your moderator for today's discussion. Before we get started, though, I would like to give an introduction to CAS first and what we do and who we are. CAS is an organization that specializes in scientific information and solutions. We are here to help you advance your scientific research and innovation. Our scientific content covers much more than just chemistry. As you can see here, we cover other areas that converge on chemistry, such as biology, human health, and material science, including things such as structure-activity relationships, analytical methods and protocols, organometallics and inorganics, among many others. Our content covers both journals and pens, and one of the best features about CAS is our hundreds of scientists and professionals like myself who work behind the scenes to curate and connect all of this data. So without further ado, let's get started. This is the third year that we've developed our Trends to Watch list, and we have been really pleased with how popular it has become. Our scientists, researchers, writers, and data professionals here at CAS spend all year analyzing, organizing, and optimizing millions of articles for use in our products like SciFinder and BioFinder. Those activities also position us well to dip into the CAS content collection to identify issues and topics for articles that will be of interest to our CAS Insights readers. If you haven't already, I highly recommend that you subscribe, and we will put that link into the chat. This year's article highlights 8 key scientific trends that are set to influence research and innovation in 2025. These trends cover a broad spectrum, showcasing the complex, interconnected nature of science that we all know so well. We look at the importance of good data within artificial intelligence. Also, the exciting progress being made in precision medicine, which aims to customize healthcare based on individual patients' genetic profiles. Innovations in battery technology are also another highlight, promising advancements in sustainable energy solutions. We also touch on developments in material science, environmental sustainability, and biotechnology. In each of these cases, we wanted to provide a quick pointer to areas with the potential to make substantial impacts. After that, it is up to our readers to go further and dig deeper into these areas that they find most exciting, which is exactly what we are going to do today with our guest panel. So we will start with Dr. Erin Streets. Erin is a professor of engineering in UC Berkeley and an associate professor in the departments of bioengineering, biophysics, and in the Center of Computational Biology. He is also an investigator at the Chan Zuckerberg BioHub. His lab develops new hardware tools, molecular tools, and software tools to study genome regulation at the single cell and single molecule level. Next, Dr. Youngtao Liu. Youngtao is on the research and development staff at the Center for Nanophase Materials Science at Oak Ridge National Laboratory. His work focuses on integrating artificial intelligence with physics to develop autonomous experimentation platforms, such as autonomous microscopy, that accelerate progress in materials science. And lastly, Dr. Shahar Keenan. Shahar combines her pioneering work in advancing drug discovery with an extensive background as co-founder and CEO of Polaris Quantum Biotech. She has held research positions at Northwestern and Duke universities and has also served as CSO for Cloud Pharmaceuticals. On behalf of CAS, welcome to you all. Thank you so much for joining us and being here with us today. First, we want to take a few minutes for each of our participants to give a brief overview of their work to help provide some context for our discussion today. We will start with Erin and then go to Youngtao and Shahar. Erin, please take it away.
Great. Thanks, Janet. Thanks for the introduction, and thanks for hosting this webinar this morning. I'm excited to join. So as Janet mentioned, my lab develops new tools to study biology at the single cell level And to give some context for how we approach our research, when you think about the technological innovations that have fueled the computing age, the silicon manufacturing industry and microfabrication, nanofabrication broadly, I think are some of the most important sort of industrial arcs that have really driven our ability to make advances in computing. Our ability to make chips that are smaller, cheaper, and have a higher density of transistors and other electronic components packed onto those chips has been the engine, sort of the underlying foundational technological arc that has made our progress from the ENIAC, the first electronic numerical integrator and computer, to where we are now, where we all have much more powerful computers in our pockets with our cell phones. And we also now have these, large computing clusters or server farms that are really powering our analysis of big data. And so in my lab, we sort of take the lessons from the silicon manufacturing industry and from microfabrication, both metaphorically and literally, and try to apply those techniques and those concepts to drive innovation in biology. And specifically, we develop microfluidic devices for multimodal single cell analysis. The idea here is that if we can pack more biological reactors onto a chip, if we can make more measurements per unit area, per unit time, and per unit dollar on cells, then we can start to approach the generation of big biological data at a scale that can really take advantage of the computing power that we have today. And so to this end, we make microfluidic devices that can perform genomic and optical analysis on single cells. And we really take advantage of DNA sequencers, which has been another field that has benefited from microfabrication, the ability to sequence more DNA per unit area, per unit time, per unit dollar has really driven this genome revolution over the past 20 years. And I think we're at a sweet spot now where we can generate data at the genome scale that we can really take advantage of the power of AI and other machine learning techniques to understand more about our genome and our cells in our body. So I'll stop there and we can talk more as we go into the panel.
Thank you, Aaron, for sharing your work. It's so fascinating to know that we can examine such small entities. Young Tao, if you'll join us back on camera and share a little bit about your work, please.
Morning and good morning, good afternoon, and good evening, everyone, depending on where you are. And my name is Yuntao Liu, and it's a truly honor for me to be here with all of you today. I'm currently a research and development staff scientist at the Center for Nanophase Material Science, Oak Ridge National Lab. And my background is chemistry and material science. So my bachelor degree is in chemistry and PhD is in material science. And I have spent past few years focusing on the application of artificial intelligence and machine learning in material science and chemistry. So one area of my research is autonomous experiments and a self-driving lab. So for example, we can leverage the artificial intelligence to accelerate the experimental process, and we can use the machine learning algorithms such as the Bayesian optimization and the reinforcement learning to guide the experimental process. So this algorithm can analyze the real-time experimental data and predict the outcomes and make the decision and identify the optimal experimental condition for the next experiment. So the robotic system and then can take this prediction to execute the experiment and control the equipment, collecting more data. So this is a closed-loop feedback system that allows the artificial intelligence to continuously analyze the data and make real-time adjustment of the experiment to achieve the material optimization and the material discovery process. So this dynamic process, we expect it can accelerate the discovery and enhance the efficiency and enable the exploration of complex material space that would be challenging to if we use a traditional experimental method. The goal here is to make the research more efficient and more effectively to unlock the new insights that would take much longer by the traditional method. So a big part of my work here is the interdisciplinary. So we bridge together artificial intelligence, advanced material synthesis, characterization, fabrication techniques, and also incorporate human expertise and prior knowledge to develop new tools and methods that can help researchers to tackle complex challenges more efficiently and more effectively. So I'm particularly excited about how this technology can create new opportunities to bridge the gaps between different fields and pushing the boundary of what we can achieve in material science. And in terms of material, so in addition to the autonomous experiment, I'm also working on the functional materials such as ferroelectric, photovoltaic, and optoelectronic material. So my work is about the advancing both the research tools and the functional material science to accelerate the progress in material research. So I'm looking forward to see how this effort can contribute to the field in 2025, and excited to be here today, and looking forward to share more about our work. Yeah, thank you.
Yes, thank you, Young Tao, for that glimpse into autonomous labs. Very exciting to think about the future for materials science there. Thank you so much. And Shahar, if you will join us real quick and share with us. Yes, the floor is all yours. Thank you.
Thanks. And thank you guys for inviting me over. This is a really interesting panel, and I'm looking forward to learning about other emerging and exciting technologies. So my background is a computational chemist, and I was always interested in looking at large chemical spaces, in finding molecules that are not the first thing that you would look up or that you would think of. When I did my PhD, the size of chemical spaces were in the millions. When I did my postdoc, it was in the billions, and it still wasn't large enough or big enough to find the right molecules. What we used to do is find molecules and then go to the lab, synthesize those molecules, and slowly increase the area around those molecules, find small changes in those molecules that would get us to where we wanted to go. This is time consuming and not very efficient. So 5 years ago or so, I was looking into how to implement an even bigger chemical space in the areas of drug design. So how to cut down the amount of time you do in just finding that drug molecule. And drug molecules are interesting because you need to have several properties of those molecules. It's not enough that they have the right biological activity, which is what the drug does, but it also has a lot of other properties around that are crucial for this being a good drug. Everything from solubility, permeability, so that the drug that you swallow will get into the bloodstream. Of course, toxicity, how stable is that drug in the body? And all of those things create a very complex set of properties. And we're looking for those molecules. We want to find those molecules that have those properties and the biological activity as fast as we can. And at that point, when we were looking at that, we realized that there's a technology that can help that, and that's quantum computing, specifically quantum annealing. It's a technology that's available today, and that you can use it to solve large optimization problems. And then we realized that drug design is really an optimization problem. And if we can think of that problem a bit differently, so instead of saying, okay, I have a molecule, how do I change it to find the best properties, the best profile? How, instead of doing all of those set of small changes, what we're doing instead is saying, okay, let's look at all possible molecules and then find the best ones. We're still not there, we're working on it, but we are looking at very large chemical spaces. The chemical space that we look at right now is 10 to the power of 30 most of the industry is looking at much smaller chemical spaces, maybe billions, which is 10 to the power of 10. So we're looking at much larger chemical spaces and finding the right molecules. And we're using quantum annealers for that. The quantum annealer that we use is by a company called D-Wave. It's an American company. It's a publicly traded company. The quantum annealer that we use have 5,000 qubits, which of course is good enough for 10 to the power 30 molecules. We want to grow that chemical space, and we work with them on increasing that chemical space and waiting to get more and more molecules, bigger spaces, and of course, bigger quantum computers. This year is going to be the year of the quantum, and I am looking forward to talking with you about that, and of course, to all the excitement in that field.
Thank you, Shahar. Yes, we are all so excited about quantum computing. It has come up a lot, I know, just in even like the regular news. So looking forward to hearing more from you and just a bit on that. Absolutely. Now, for what everyone has been waiting for, if all of our speakers can come back on camera, we will start our panel discussion. And let me first ask each of you, which of the trends covered in our CAS article jumped out at you most? Either because it was something that you were expecting to see, you're in agreement with it, or maybe it's something that you weren't expecting to see. Erin, we will start with you, and then we'll go to Young Tao and Shahar. So Erin, which trend spoke to you the most when you looked at them?
Thanks, Janet. Well, I mean, the last one, Omniomics, the next single cell revolution, is right smack in the middle of what we're interested in. And I'm not surprised to see it, but I'm happy to see that this is identified sort of beyond the field of biology and bioengineering as an important trend. I would say that to kind of add to that, there's a lot of activity in this space, and I think a fraction of it is really important for understanding biology. And then because it's such a hot space, there's a lot of development of tools and a lot of development of software. And I think trying to make the development of those new technologies more impactful is something that we have to work on. I think to that end, my sort of broad answer to your question is that the combination of the omniomics and the trend titled data quality emerging as a key driver for AI success really go hand in hand. I think that in the field of single cell analysis, it can be easy to not really use the right analysis or not to have the right kind of data for the questions you're asking. And I think as we move towards larger multi-omic data sets that are going to be evaluated and analyzed by generative models, it's important to design our data capture techniques in a way that are matched with the way that the analysis is designed. And I think we're going to have an opportunity to do that in the next year.
Oh, that is great. And we will talk actually more on those exact topics in just a few minutes. So awesome. Thank you so much. Young Tao, which trend stood out to you the most?
I think what grabs my attention is also data quality emerging as a key driver for AI success. So I think this data quality highlight a fundamental principle in the field of artificial intelligence and machine learning. So the quality of the outcomes produced by the machine learning model is largely dependent on the quality of the input data. I guess this is in particularly important in scientific research. So the data quality, when it comes to the artificial intelligence reliability. So the high quality data can ensure the AI model developed for scientific purpose are more accurate and also more robust. So for scientists, the fidelity of the data often impact the credibility and the reliability of the research result. So and another thing is when we talk about data quality in scientific discovery, so sometimes the outlier data, so usually we treat them as a low quality data, but in scientific research, this outlier data might mean new physicals. So we also need to think about how we define the data quality in scientific research. So I think it's very important to control data quality in scientific research, and in particular when we use artificial intelligence. And also, I want to, since my research field is include a self-driving lab and autonomous experiment. So I think this kind of technology are transformative technology in the field of material science, and it can also help us to generate a large amount of high quality data that can be useful for training the artificial intelligence model for scientific research. So the key advantages in self-driving lab can include it can operate 24 hours and 7 days a week, and it can reduce the human error, and it can explore a broad range of experimental conditions, and this broad range of experimental conditions can help us to generate a very large amount of high quality data. So this high quality data we can exclude the human bias and from this data, and this can potentially lead to the novel material discovery with unique property as well. So however, there are also challenges in the self-driving lab, such as high initial cost in building the self-driving lab, and the complexity in system integration, and also the data management issue. So I see nevertheless, I see a beautiful future, a promising future for combining self-driving lab with artificial intelligence, not just using artificial intelligence in building self-driving lab, but also using the self-driving lab to generate more useful data for training useful artificial intelligence model. Yeah, I will stop here.
Yeah, some great points you made there about high quality data, and we will definitely touch on that in just a few minutes as well. So Shahar, which trend did you find most interesting?
So one of the reasons when we started Polaris is we wanted to be able to produce drugs for as many diseases as possible. And I think the combination of CRISPR and single cell technologies are increasing our understanding of diseases in a way that personalized medicine can become a much more achievable goal. When we use CRISPR to understand single cells, we use the Omniomics to understand the biology inside those cells. We can understand which target it is that cause a disease and develop drugs for that. And again, if we can do that using the right technology, collect the right data, right, and combine these together, I think that's when we can start talking about personalized medicine, what's relevant for you, what's relevant for the person standing next to you, and how your different biology change what's the right molecule that will change your disease progression. And I think the combination of those, so personalized medicine, CRISPR, Omnionics, and of course, the right data and cleaning the data in the right way, these together can combine to bring us a better future. And I was really happy to see all of those things together on your list.
Yes, that you're speaking to my heart. A lot of my work this year was dedicated to helping build a database based on bioactivity. So all of those things you talked about are just so important. And personalized medicine, we couldn't ask for anything better to think about for the future. So we all have talked about high quality data. So next, let's discuss artificial intelligence. No matter what field we are in, we have been hearing this term more and more as time goes on. And the CAS Insights article specifically talked about how high quality specific data sets will be that key driver for successful AI moving forward. And I think all three of your work here touches on machine learning, but within very different scientific disciplines. So I'll start with Young Tao, and then I'll ask Shahar and Aaron just to give us any more thoughts that you might have on that high quality specific data sets being the key driver for successful AI-driven scientific experiments going forward? Young Tao, if you'll start us off.
Yeah, thanks. So yeah, I think we all discussed about high quality data, and I guess we all agree that high quality data is important for training robust AI model. Yeah, I also talked about self-driving lab just now. I want to talk about extend the topic a bit more to the accessibility of high quality data. So when I talk about self-driving lab, I mentioned some challenges in building this self-driving lab. So it's expensive and it's complex to build a self-driving lab, even if it can generate high quality, large amount of scientific data. So this poses a question that is the data from self-driving lab is potentially not accessible to every machine learning or artificial intelligence scientist. So, so it's crucial to enhance the sharing of high quality data under some policies such as the FAIR policy. So this principle, the FAIR principle, aims to ensure the data and are easy to find and are accessible under well-defined condition and is capable of integration with other data sets and can be reused beyond the original purpose of what they were collected. So this widely accessible high-quality data can provide a solid foundation of reliable data sets that for both the researcher in both academic and commercial institutes. So this accessibility allows both the individual researcher and the researcher from small institution who has the limited access to the large self-driving lab to engage in the artificial intelligence project without the barrier of data acquisition cost. So yeah, so I think the accessibility of the data, high quality data, can also ensure the improvement in AI technology.
Thank you for bringing up that point about accessibility and the importance of fluidity and sharing of the data that we have. Shahar?
Yeah, so the area that I'm in computationally is drug design. And one of the issues there is that there's not a lot of data. The data is not very clean. It's most things fail. So the data sets are not well balanced. And a lot of the data is collected by organizations like CAS from many publications. So different measurements, different quality of data. And I think for us and for the industry, being able to talk about the quality of the data, the availability of it, balancing it, is something that's crucial to be able to continue to build those capabilities. We worked with one data set that had a couple of thousands of molecules in it. In chemistry, this is a really large data set. I'm sure that in other fields, biology or self-driving car, a couple of thousands of data points is really a small set, probably not well balanced as well. So for chemistry, for drug design, the ability to look at data, to clean the data, and to start bringing in more data, I think is going to be crucial this year. And I definitely support Young Tao here. It's some of that data is in silos. Some of that data is not publicly available. And the more publicly available that we make that data, the more access for researchers to get to that data is going to make the whole field better.
Great. Thank you for adding to that about, just again, reinforcing the accessibility of data and then also pointing out just how data sets, how much work is involved in cleaning that data to make sure that you have accurate data for your models. Great points. Aaron, please go ahead.
Sure. Yeah, and actually, yeah, as Shahar pointed out, in terms of thinking about the size of data, I think biology is really one of the biggest big data challenges that we're confronted with these days. If you think about it, every cell in our body has 2 copies of our genome. Our genome has 3 billion base pairs. And we have, in each cell, not only has 2 copies of those 3 billion A, T, C's, and G's, but also then transcribes that genome to produce transcripts that represent tens of thousands of different genes that then can get translated into hundreds of thousands of different proteoforms. And so if you think about how much data is just in one cell, now imagine that each cell in our body is different, and we have trillions of cells in our body, and then there's 8 billion genomes, humans, around the world. And so one of the promises of AI in biology is to develop a sort of a generative model that can predict the impact of genomic or molecular perturbations on cells, so that we can understand how mutations might lead to disease, or how it might disrupt development, or how we might engineer new biological systems in the future. But to make a model that is trained on such a large, vast, and diverse amount of data, this is really an international effort. And so imagine something like the Human Cell Atlas Project, which is a large international consortium whose goal is to essentially map all the different cell types in our body across genomic variation, so that we could, in theory, train such a model. Well, in order to do this well, we need good sampling of the genetic diversity of our species, which means that we need to have sort of unbiased sampling. We need to collect, genetic diversity from, all people across the world with some frequency. And when we then share that data, as Youngtao pointed out, we need to collect that data in such a way that we can compare between labs and between experiments without confounding with the technical variation that might occur from the way that experiment was done over here versus the way that an experiment was done over there. Right now, we spend a lot of time and effort developing new computational tools that can manage the technical variation across data collection sites before we can even start to ask questions about the real underlying biological variation. And so I think, to both Youngtao and Shahar's point, if we want if we envision a future where AI can really impact human health, we need sort of structured data collection pipelines, and then that data, as Youngtao pointed out, needs to be accessible to academics, and in such a way that we can easily develop algorithms that can scrape that data for academic research. And so that's some of the big challenges going forward.
Thank you for adding those challenges of the data standardization and data structure, and how important that is across disciplines. After AI, the technology that probably gets the most discussion across disciplines these days is quantum computing. So Shahar, you told us a bit about your work you're doing at Polaris Quantum Biotech specific to drug discovery. Do you think that the work being done there has implications across other scientific disciplines? And are there types of research that you think might inherently be better or worse for quantum computing?
So I'll start from our small corner of the world, which is what we do is molecular design, molecular optimization. Of course, that has a lot of implications, not just in drug design, but in agricultural, in material science, and other fields where you need the right molecules. The computers that we work with are quantum annealers. Quantum annealers are really good at solving large optimization problems. Everything from supply chain optimization to figuring out more efficient ways to run the port of Los Angeles or to put boxes into airplanes. All of these are large optimization problems that people are now using quantum annealers to solve. A lot of it got to do with balancing things, balancing portfolio, especially for if you're thinking about commercial or fintech applications. Quantum computers themselves can solve problems that are going to be very difficult for either CPU-based computers or GPU-based computers to solve. However, they are only efficient in solving very limited number of problems. So if you're lucky and your problem can be solved on a quantum computer, that means that you can enhance your timeline. You can solve those problems much faster. We will not have a quantum computer that runs Word or WebEx application. However, we will have quantum computers that solve factoring of two molecules, and that means that there's a problem with our current crypto and cryptography solutions. All of our passwords are protected in a way that is right now challenged by quantum computers. There are other things that you can solve on a quantum computer that are very efficient in solving those problems. But it's going to be a while before they're ready. I'm guessing 3 to 5 years if you're not solving an optimization problem.
Yeah, so it seems like there are many different fields that it could help, but also lots of different fields and room for improvement that are not currently being utilized. So that's very encouraging. Aaron, moving on to you, one of the trends that we did note, and you have commented on obviously, was the combination of the single cell applications and the multi and omniomics. What does this kind of multi-layering of the single cell analysis and therapy, how does this provide additional benefits?
Certainly. So when we think about the current state of single cell genomic analysis, one of the goals, as I mentioned, is to sort of quantify the identity of all the cell states that our genome can encode. And one of the ways that we've done this significantly over the past 5 years is through high throughput single cell RNA sequencing, where in a single experiment, we can measure the transcriptome, so we can count the hundreds of thousands of transcripts in individual cells at about up to 10,000 cells in a single experiment. And so what this does is it allows us to sort of cluster cells by their gene expression and define different cell states sort of algorithmically by looking at variation in the transcripts that they contain. But looking at gene expression is only one lens through which we can identify the identity of a cell. Because as we mentioned, every cell has the same genome, but they're kind of performing their functions and they're reading different parts of that genome differently. And so other information like the proteome of a cell, the physical morphology of that cell, its epigenetic landscape, these are all important layers of molecular information that we can use to increase the precision or the resolution of our cell atlas, of our atlas of all the cell states. And so one way that people think about multiomics is by sort of increasing the fine-grained definitions of different cell states by layering on simultaneously other molecular analysis. But another way to think about single-cell multiomics is instead of thinking about it as a definition of cell states, by being able to measure multiple layers of molecular information in single cells at high throughput, we can start to understand the information flow between those molecular layers. So for example, we can understand how variation in the genome or the epigenome impacts variation in gene expression, and how variation in the proteome might then play back in on gene regulation and then the translation of other proteins. And so with enough data, we can start to build a model, as I mentioned, that can look at information flow in the central dogma, DNA to RNA to protein, through the lens of different cell states. And in this way, we can start to ask questions about how we can make perturbations in certain types of cells, like immune cells, that might impact their function. How can we design a better CAR T? Or how can we understand the impact of genetic variation in non-regulatory and non-coding regions on health or disease? And so I think there's a lot of potential in this field moving forward.
Yeah, it'll be interesting to see how that holistic view helps insights and new applications. It'll be very interesting to watch. Thank you.
Of course.
Young Tao, one of the trends that we covered broadly was the wide variety of innovations coming out of material sciences. We didn't specifically get into ferroelectric materials, but my understanding is that they have also a surprisingly wide range of applications. Can you tell us a little bit more of about those applications, since you mentioned you work with the ferroelectric materials?
Yeah, sure. So yeah, it's indeed have a very broad range of application. It's a very old material. The history of ferroelectric is about over 100 years. So they are very interesting material because of unique electrical properties. So ferroelectric material can spontaneously develop electric field polarization, and you can flip this polarization back and forth using an external electric field. So this kind of feature opens up a lot of application in different field. So for example, this kind of, this switchable polarization can be used for making the memory device, such as the ferroelectric random access memory. And it is great because this kind of memory can keep data without needing the power. So, it uses less power than regular memory. And also all ferroelectric material at the same time are also piezoelectric material. So thanks to these piezoelectric properties, ferroelectric material can also be used in sensor technology. They can turn the mechanical energy into the electrical energy, and also turn the electrical energy to the mechanical energy. So this makes them a perfect material for sensing technology. And you can use the sensor to pick up the pressure or the movement and change it to the electrical signal and capture it. And also this kind of feature also lead to their application in energy harvesting application. So we can turn the pressure from, for example, from the footsteps into electrical energy. So you can think about the power. You can power in the light, turn on the light just by working on them. And it also has an optical application and a medical application. So in the medical world, the ferroelectric material can help make ultrasound probe that doctor can use this kind of device to look inside your body without having to cut you open. So I'm just joking here. Yeah, so it has a lot of application. I guess I cannot cover all here today. And the researcher and the scientists are finding more and more new application of ferroelectric material. So yeah, that's it.
That is a great segue into our next question, which will be on green energy. So thank you. Before moving to the audience questions, I do want to ask this very last broader question about green science in general. 3 of the trends we covered on the CAS Insights article were battery innovation, material science applications, and waste management within the circular economy. Deal directly with that ecological issues. So how much of a consideration is this in all of your work, and how should we all incorporate this reduce and reuse thinking into our research and development? I'll start with Shahar and then go to Aaron and Young Tao. Shahar, any thoughts?
So one of the good things about using computers is that you don't need to do the experiment yourself. So you're saving time and people's time and even just driving to the lab, all the materials, all the environmental impacts of running a lab. For us using quantum computers, they are faster. And that means that even, if you need to do some simulation and it takes you a month of computer time, if you can run it on a quantum computer and it takes you a minute, you saved so much time, just energy, electricity. Being on the computational side, we see this, I think, through how can we save time and waste by running things on the computer and find the fastest computer to do that.
Great points. Excellent points there. Yeah. Thank you so much, Aaron.
Over the past, let's say 5-10 years, actually 20, if you think about since the first DNA sequencers, there's been a lot of data collected already. Sometimes I think more than we really have the time or energy to analyze. And this data generation means a lot of experiments, a lot of consumables, a lot of plastics, a lot of buffers, a lot of people's time, as Shahar pointed out. And I think we kind of had to go through that to learn how to make these measurements. But I think in the future, it's important to be a lot more efficient with the data that we collect, because with this huge data set that we've already generated, just the publicly accessible genomic data that's out there right now, for example, on NCBI or something, not only is it a lot of expended resources, but it also takes a lot of computational power to sift through all that data. And if we can be more efficient about the way that we collect data, it not only impacts our use, our materials consumption, but it also will minimize the amount of computational power that we need to sift through and gain insight from that data. So I think this is a really important question.
Yeah, I love how you tied that back into the data standardization and structure. Like if we can, solve that problem with the get-go, we can save so much on the end here. Young Tao, any more thoughts on this green cycle here we're speaking on?
Yeah, I support Shahar and Aaron's point about, we can consider to use less energy to do research, to discover new material. And one of the research topic I engaged in is the highlight perovskite. So I want to share a story about the highlight perovskite, which is the photovoltaic material used in solar panel technology. So, this material their outstanding property can allow them to achieve a very high power conversion efficiency. The beauty of the highlight perovskite is not only in their outstanding photovoltaic performance, but also in their low production cost and energy requirement. So they can be processed at low temperature and through solution-based method, such as preheating or coating. So this kind of processing significantly reduce the environmental impact compared to other like high temperature processing of silicon. So, and also to commercialize the perovskite solar cell, the researcher is also considering the stability and the toxicity of the lead-based perovskite. So ongoing research activity is including in finding material with less lead in the highlight perovskite material to seek to develop non-toxic alternative and also enhance the long-term stability of perovskite solar cell. So this story, the reason I want to share this story is like this story highlights the importance of considering the entire life cycle of a material, not just the material itself. It also includes the fabrication process of the material and the application of the material to determine its overall environmental impact and the sustainability. So all these efforts, so making material itself and its application are crucial for the, for the environmental consideration.
Yeah, thank you for sharing that insight on the manufacturing aspect for material science as well. That's great. Well, thank you all so much for all of these insights. I know this was a ton to cover in such a short amount of time we have here today. I do want to make sure we get in some of these audience questions. So our first audience question to Young Tao, as someone with no experience in AI machine learning, what recommendation would you give to them for getting started with machine learning in their research?
Yeah, so the machine learning, the machine learning is, largely Python driven. So the Python is a programming language. So the first step is to learn the basic Python programming if you are not already familiar with it. And in this learning process, I would recommend focusing on understanding the concepts such as data type and loops, functions, and libraries like the NumPy, Pandas. And these libraries are very important for data manipulation in machine learning field. And also the next is, instead of focused on a specific machine learning algorithm, I think it's important to understand the fundamentals of machine learning. So take some time to learn the broader concept, such as what is supervised machine learning, what is unsupervised, and what is the model evaluation, what is overfitting and underfitting. So you can use some online platform. There are many excellent introductory courses. So and which can provide a very solid foundation about some machine learning algorithm. And once you get this understanding, you can start to use the hands-on machine learning, use some hands-on machine learning tools, online machine learning tools to analyze your data, and then you can deepen your understanding through using the machine learning. And so it's very important. The practice is very important. So you apply your knowledge to small projects and analyze different data sets, and then solidify your understanding and also help you develop problem-solving skills that are more critical for the advanced machine learning work. Yeah, that's it.
Yeah, thank you for those tips. I know we have a lot of early career scientists joining us today, and many of us are not familiar with getting AI into our workloads. So thank you for those tips. Next, Shahar, someone has asked, can you discuss targeted drug design for specific diseases a bit more for them?
Sure. I'm not sure I understand what is the targeted drug design, but if they are thinking about, personalized medicine, then the question is, in many cases, if we understand disease, we know how to stop disease progression. And in many cases, that means stopping a very certain protein activity. So proteins are the cells machines. And if you understand the pathway that's relevant for your disease, then you understand which proteins are relevant for that pathway. And then you know how, if you stop a certain protein, you may be able to change the disease progression. It's complicated because you don't want to stop a protein that's relevant for other processes in the cell. But when you have a protein that is a target, and everything I described so far is biology, and understanding the work of a single protein in a cell is a complicated process. Okay, I don't, and Aaron probably can tell us much more about those things. But when you have a protein that you are interested in stopping the activity, that's when we come in. That's when we find a small molecule or a peptide, or even an antibody that will block the activity of that protein. That you can think of it, there's a big machine and you throw a wrench into it. You stopped the activity of that specific part of the machine and the whole machine stops. And that's where we come in. I'm not sure if I answered the question or not.
No, that's great. Yes, I am sure. That's absolutely wonderful. Yes, we all always can use a bit background on how our medicines actually do work. They're actually looking for those targets to help not only cure diseases, but relieve symptoms for sure. So Aaron, one question has come up. I'm going to open this up to you, and then anyone else, of course, please comment if you have anything. But does the AI-based experiment give proper results, as laboratory-based experiments give exact and real experience? Can you speak a little bit on that, please?
Yeah, this is a good question because there's been a lot of discussion in the literature over the past couple of months about the notion of building a virtual cell or a foundation model of the genome, ways that we can, maybe with the data that we have now, or maybe on data that we're collecting, develop a model that can predict, that can sort of generate the outcome of molecular perturbations in a way that we could, in theory, then do experiments in silico instead of doing those experiments on cells. And I think, I mean, this seems like science fiction. It certainly would have a couple years ago. Now it seems like something that we might be able to do in a few years, and we already are making iterative models that can predict the presence of proteins in cells that we didn't measure those proteins. But I think that this is a good question because this all comes back to the question of data quality. Like the accuracy of these predictions is only going to be as good as the sort of combination of the algorithms that we're using and the data that we're training this on. And if we're missing important aspects of genetic variation, if we're not sampling a certain genetic population of our species, or if we're not looking at a certain type of cell, then we're not going to get the right answers. And so I think it's going to be a constant push and pull between testing the accuracy of some of these generative models, and then doing more experiments to validate their results. And I think this is going to be a continuing endeavor as we merge these two fields. It's a great question.
Yeah, lots coming back to the data quality. Absolutely. It's lots of different discussion today, but it all seems to stem back to if we don't have great data, then our results won't be so great. So one last question for everyone. Let's talk a little bit about the interdisciplinary science in these trends. So if everyone would just like to speak a little bit before we close out the webinar on this, that would be great.
Sure. I mean, this is a great point. I'm trained as a physicist and engineer, and in order for our work to be impactful at all, it's really important that I work closely with computer scientists and biologists, immunologists, virologists, not only to develop our tools and to analyze our data, but to gain insight into biology. And I think, whereas maybe science of the past were these sort of kingdoms of individuals who are experts in their field making new discoveries, I think the science of the future is really about teams of people with different expertise tackling much larger problems that we've been talking about today.
Absolutely. Shahar, Yungtao, any last thoughts?
I want to add one more thing, and that's besides being versed in multiple areas of science, I think scientists are also or should be versed in how to present their work. And that's one of the trends that I see a lot lately, is asking scientists to participate in the discussion, not just of what does their science do, but what is it good for, what's the cost of it. And I think that's something that we're going to see much more in the upcoming years, is the participation of scientists in that discussion, which I think is really important to be able to present what you do and to explain to others an experiment, how good it is, and what's the role of that experiment in the whole field of it.
Well, thank you so much for joining us today in this conversation, for accepting our invitation. That looks like it's going to be our last question as we've come up on the hour. Many thanks to Aaron, Yungtao, and Shahar for sharing your time and, most importantly, your expertise with us. Are there any quick final thoughts you would like to leave with us before we sign off? We can start with Shahar.
Sure. So this year is going to be the year of quantum. Join us, learn about quantum. It's an exciting field. Things are changing on almost on a daily basis. And it looks like it's going to be a great year. So dig in, people, dig in.
Yungtao.
Yeah, so thank you all for joining us today. So I think we discussed a lot of topics today. So from the quantum computing, nanoscience, drug discovery, and AI. So I'm truly excited about the potential of artificial intelligence in material science and in nanoscience. So I think by combining the human expertise with the efficiency of machine learning and AI, we can potentially unlock new discovery and address some of the most pressing challenge facing our world today. So, and I'm looking forward to see how our effort can contribute to the field in 2025. Yeah, thank you again for your time.
Aaron.
Thanks so much for having me. I think that the trends that we covered today are certainly some of the most important trends to watch in 2025. But I think one of the most exciting things about science right now is that with this interface between data science and computing and all of the topics that we talked about today, some of the most exciting things that happen in 2025 are going to be really hard to predict. I don't think I could have told you in 2023 what my favorite papers from 2024 were going to be. And so it's really exciting to see what this interface of interdisciplinary science is going to produce over the next few years. But thank you so much for having me, and it's really a pleasure to be among the panelists today.
Yes, and for my final thoughts, of course, I would like to thank everyone again for joining us today and for all of your wonderful questions. It's so great to be able to get these audience questions answered. I would like to encourage everyone to please come and visit CAS Insights and subscribe if you have not. We have many other scientific topics to explore there at the intersection of science, technology, and innovation. Thank you again for participating in this webinar today, and we will see you next time. Bye, everyone.